Welcome to this lecture on reproductive biology. Today, we are going to explore one of the most magical and critical steps in human reproduction, implantation. We will understand how a tiny cluster of cells communicates with the mother's body to establish a pregnancy, and how doctors use mathematics to track this incredible journey. To begin our journey, imagine trying to plant a delicate seed. If you throw it on hard dirt, it will not grow. The soil must be soft and nutrient-rich. In human reproduction, the embryo is our seed, and the mother's uterine lining is the soil. Implantation is the highly coordinated event where this tiny embryo buries itself safely into the prepared uterine wall. This is not just a physical collision, it is a complex chemical conversation between the mother and the embryo. Without this perfect docking process, a pregnancy simply cannot begin. Now that we understand what implantation is conceptually, let us look at the structure of the embryo when it arrives at the uterus. At this stage, the embryo is called a blastocyst. It is not just a random ball of cells. It has two distinct parts. The outer layer of cells is called the trophoblast. This layer is crucial because it will eventually develop into the placenta, the life support system. Inside, there is an inner cell mass, which will grow and fold to become the fetus itself. Both parts play vital roles in the upcoming attachment process. Knowing the structure of the blastocyst, we can now explore how it actually attaches. Implantation occurs in three distinct sequential stages. First is apposition, where the embryo simply rests loosely against the uterine wall, like a boat gently touching a dock. Next is adhesion, where specialized proteins act like anchors, firmly attaching the embryo to the tissue. Finally, we have invasion. Here, the embryo literally burrows into the maternal tissue. The outer trophoblast cells secrete powerful enzymes that break down the mother's tissue, allowing the embryo to tap directly into her blood supply for nutrients. Before we move on to the timing of this process, let us see this with a concrete visual diagram. On the screen, you can see the three stages we just discussed, moving from left to right. On the far left, during apposition, the blastocyst is just making contact with the ciliated epithelial cells of the endometrium. In the middle, during adhesion, notice how the outer trophoblast cells are gripping the microvilli tightly. Finally, on the right, during invasion, the syncytiotrophoblast has penetrated deep into the endometrial stroma, reaching out to connect with the red maternal blood vessels. This establishes the vital feto-placental unit. Having seen how the embryo burrows into the tissue, we must now ask, can this happen at any time? The answer is no. Implantation is the primary bottleneck in human reproduction. The uterus is not always welcoming. It is only receptive during a very brief period known as the window of implantation. The endometrial lining must be perfectly primed by hormones like estrogen and progesterone. If the embryo arrives too early or too late, it will be rejected. This strict biological timeframe is the absolute cornerstone of modern fertility treatments like in vitro fertilization. This biological window leads us to an important mathematical relationship used by doctors. The formula for the window of implantation is WIMP equals D-ov plus delta D. Let us break down what each symbol means. WIMP represents the specific days of the menstrual cycle when the uterus is receptive. D-ov stands for the exact day of ovulation, when the egg is released. Finally, delta D represents the time delay required for the fertilized egg to travel down the fallopian tube and mature. In humans, this delay is typically between 6 and 10 days. If the delay increases beyond this, the window shuts and attachment fails. Now that we have our formula, let us apply it to a real-world clinical scenario. Suppose a patient at a fertility clinic ovulates exactly on day 14 of her menstrual cycle. We need to find her receptive window. We know the biological delay is between 6 and 10 days. First, we calculate the earliest possible day by adding the minimum delay of 6 to 14, giving us day 20. Next, we calculate the closing of the window by adding the maximum delay of 10 to 14, giving us day 24. Therefore, the optimal window for this patient is strictly between day 20 and day 24. Outside this tight time frame, an embryo will almost certainly fail to implant. So, we have successfully timed the implantation, and the embryo has attached. What happens next? Once the embryo invades the endometrium, its outer trophoblast cells immediately begin shouting for survival by secreting a hormone called human chorionic gonadotropin, or HCG. This specific hormone is absolutely crucial. It travels to the ovary and rescues a structure called the corpus luteum. By doing so, it ensures a continuous production of progesterone, which maintains the thick, nutrient-rich uterine lining. Without this vital hormonal signal, the lining would shed, and the pregnancy would be tragically lost. Understanding the vital role of HCG, we can see why medical professionals monitor it so closely. Because the embryonic cells are multiplying rapidly, the amount of HCG they secrete grows exponentially. Doctors track this exponential rise in the mother's blood to confirm that the implantation is healthy and ongoing. If the hormone levels are rising too slowly, it is a major clinical warning sign. A slow rise strongly indicates either a failing implantation or a dangerous ectopic pregnancy, where the embryo has implanted outside the uterus. This makes tracking HCG a life-saving diagnostic tool. This exponential rise we just discussed can be modeled perfectly with mathematics. The formula is C of t equals C0 times 2 raised to the power of t over td. Let us understand these variables. C of t is the expected hormone concentration at a given time. C0 is the initial concentration detected on the first blood test. The variable t is the elapsed time in hours between tests. Finally, td is the doubling time of the hormone, which is typically 48 hours in a healthy early pregnancy. If the doubling time td increases, the fraction becomes smaller, showing that the embryo is struggling to grow. Let us see this exponential growth formula in action with a clinical example. A woman has an initial HCG concentration of 50 milli-international units per milliliter. The doctor orders a follow-up test exactly 96 hours later. We assume a healthy doubling time of 48 hours. We substitute 50 for C0, 96 for T, and 48 for TD. The fraction in the exponent, 96 over 48, simplifies perfectly to 2, representing 2 full doubling cycles. 2 squared is 4, and 4 times 50 gives us 200. The expected concentration is 200. If her lab results match this, the doctor can confidently confirm the embryo is growing vigorously. We have covered a tremendous amount of ground today. Let us review the key takeaways. First, implantation is the magnificent, multi-step bridge between fertilization and a viable pregnancy, occurring through apposition, adhesion, and deep invasion. Second, this process demands incredible biological synchronization. The embryo must arrive during a very narrow window of time when the uterus is perfectly primed. Finally, once attached, the embryo secures its survival by secreting HCG, and we can use exponential growth models to track this hormone and confirm the beginnings of human life. Thank you for joining me in exploring this miraculous biological event.